Hi everyone, and welcome back to the digestive system. This is part 2. The small intestine is a narrowish diameter tube about 2.5 centimeters in diameter and approximately 3 meters long. It occupies much of the abdominal cavity. For historic rather than functional reasons, it is divided into 3 sections: the initial short duodenum, which is about 25 centimeters, the jejunum, which is approximately 1 meter, and the ileum, which is approximately 2 meters. It ends in a valve known as the ileocecal sphincter. The muscularis of the small intestine is organized into circular folds known as plicae. This greatly increases the surface area available for the absorption of food. Within these, the mucosa is organized into finger-like projections, or villi, which contain blood and lymph vessels known as lacteals and other lymphatic tissue. The columnar epithelial cells of the mucosa have microscopic projections, or microvilli, on their lumen-facing surface. Together, the villi and microvilli increase the surface area available for absorption 500-fold over a smooth tubed surface of the same size. Most of the digestion and absorption of food and water occurs within the small intestine. So these diagrams available in your textbook show the structure of a villus and microvilli. As you can see, the villi is highly vascularized with its own capillary network. Movement of chyme through the small intestine is by 2 mechanisms: segmenting waves, which push chyme to and fro against the mucosa, and slow peristaltic waves, which move chyme through over approximately 1.5 to 2 hours. A meal generally takes between 3 to 5 hours to pass completely through the small intestine. The duodenal submucosa contains duodenal glands which produce an alkaline mucus that also helps to neutralize incoming acid. Between the villi are intestinal glands, and these produce a slightly alkaline watery juice which contains some protective mucus. The digestive enzymes, which hydrolyze food molecules, have already entered the small intestine in the pancreatic juice, as discussed in the previous session. So the absorptive epithelial cells of the intestinal mucosa secrete enzymes that complete this digestion, and they do so by further hydrolyzing the resulting lower molecular weight products, especially disaccharides, by the enzymes maltase, sucrase and lactase, peptides to amino acids by peptidases, and the intestinal glands also contain hormone-secreting cells, especially for secretin and cholecystokinin, or CCK, that was mentioned in the previous session. This diagram from your textbooks shows the duodenal glands lying within the submucosa that are responsible for the production of alkaline mucus. This also helps transit of food substances through the gastrointestinal system. The products of digestion, along with inorganic ions and vitamins, are absorbed by the mucosal epithelial cells and are passed into the bloodstream and there to the liver. Larger fatty acids are in complex small globules, or chylomicrons, and these pass into the lacteals, or the lymph vessels in the villi. They bypass the liver altogether, going through the lymphatic system to be emptied straight into the bloodstream at the thoracic duct. 95% of the water in chyme is absorbed in the small intestine by osmosis. Only a litre or 2 of water per day is ingested in the diet, but several litres enter the GI system as digestive juices, and hence these need to be reabsorbed and reused to avoid rapid dehydration of the body. So, while some digestion has initially begun in the stomach, absorption in the small intestine includes sugars, amino acids, short-chain fatty acids. Long-chain fatty acids form chylomicrons, and these pass straight into the lacteals, inorganic ions and vitamins. 95% of water is absorbed in the small intestine. We'll now move on to look at the large intestine. The large intestine, or the colon, is much wider, 6.5 centimeters on average, and it lies in most of a square around the sides of the small intestine, which open into it via the ileocecal sphincter in the lower right part of the abdominal cavity. It is approximately 1.5 metres in length, and it is made up of the ascending, transverse, descending and sigmoid colon, before it ends in a short rectum and anal canal, which open to the outside via the anus. The longitudinal layer of muscularis is formed into 3 longitudinal bands of muscle, and these contract to gather the colon in a series of pouches. So this diagram from your textbooks shows the ascending, transverse and descending colon, along with the sigmoid colon, the rectum, anal canal and the anus. The large intestine ends at the anal sphincter, of which there are 2. An internal anal sphincter, which is under involuntary control, and an external anal sphincter, which is under voluntary control. The mucosa of the large intestine does not form circular folds or villi, although it does contain many deep intestinal glands, and it's lined by absorptive epithelial tissue with microvilli and goblet cells which produce mucus. It is worth comparing the structure of the mucosa at different stages in the gastrointestinal system. We can see that the gastric mucosa has small gastric pits. In comparison, the small intestine has these large villi extensions covered in microvilli, and the large intestine is relatively smooth, although it contains many deep intestinal glands. This is reflective of the degree of absorption in each of these 3 sections. Most of the remaining water is absorbed within the large intestine, so that the contents form semi-solid feces, whose passage is aided by mucus produced earlier within the gastrointestinal system. Much of the remaining digestion is carried out by the local bacterial population, which in this very anaerobic environment ferment any remaining proteins, carbohydrates and fatty acids. Sometimes this results in production of hydrogen and methane gases. Within the large intestine, bilirubin is oxidized into the more brownish stercobilin, which gives the contents its colour, and some vitamins are synthesized and absorbed into the body. Slow, strong waves of mass peristalsis push the contents into the rectum after anywhere between 3 and 10 hours in the large intestine. Stretch receptors will initiate the defecation reflex to empty the rectum. This involves the muscles around the rectum contracting to increase pressure, and parasympathetic activity causes relaxation of the internal anal sphincter. Finally, the external anal sphincter voluntarily relaxes as appropriate. Thus ends the passage of food through the gastrointestinal system. We will now consider the control of the GI system, and this occurs in 3 phases. Cephalic, or the head phase, which is feed-forward signals. The sight, smell, taste or thought of food stimulates parasympathetic nervous system via the vagus nerve, as well as the submucosal plexus. This initiates salivary gland secretion, gastric juice secretion by the gastric glands, and stomach motility in anticipation of the arrival of food, and it also stimulates pancreatic juice secretion. This impetus is maintained after the initial nervous system has died away by the hormone gastrin, and it's secreted into the blood by the stomach G cells and reaches the other gastric gland cells shortly afterwards. The next phase, or the gastric phase, is also known as autoregulation. The food bolus arrives within the stomach and is detected by stretch receptors sensitive to the volume of the stomach, and chemoreceptors sensitive to the increase in pH caused by the arrival of the alkaline bolus. These both cause stomach motility and the production of gastric juice by the gastric glands to continue. As the volume of chyme remaining in the stomach lessens, the stretch receptor and chemoreceptor activity reduces, thus reducing gastric juice secretion and motility. The third and final phase, the intestinal phase, involves feedback signals. Chyme entering the small intestine causes the secretion of 2 hormones, secretin, which reduces gastric juice secretion, and cholecystokinin, CCK, which reduces stomach emptying and promotes bile release. Both of these act to match the gastric activity with the lower capability of the small intestine. CCK also activates pancreatic juice secretion and bile release into the duodenum. In addition, the duodenum is stretched, and an extensive network of nervous reflexes in the submucosa acts as a feedback control to slow gastric activity, and a feed-forward control to stimulate both motility and secretion further down the small intestine, and to promote the opening of the sphincter into the large intestine. Digestive problems can be caused by a variety of factors, including stress, infection, low levels of stomach hydrochloric acid, lack of dietary fibre, medications, genetics, poor quality food, environmental irritants, lack of exercise, or cigarette smoking. The importance of maintaining a healthy gastrointestinal system has been known for quite some time, although obviously the means by which this is accomplished has changed somewhat. Many of your future patients, particularly those with diabetes, may be seeing a dietitian to give them advice on their diet and their health. That concludes this session on the gastrointestinal system.